Oh, yeah. Good day, everyone. Hello and welcome. Thank you for joining us for the fourth annual Black and Hymnal Week, a week where we celebrate, amplify, and support Black voices in hymnology. So, before we begin, we would like to thank our major sponsors who have made this week possible for us. And you can find the full list of our sponsors on our website. Do not forget to tweet about our section today using #pathogens and vaccines. And you would like to capitalize each word for accessibility purpose, we would have live captions and ASL interpreters during the section. You click on the CC at the bottom of the screen to access captions. You can submit questions through the comment function on the platform, not the Q&A. So as a reminder, we would require the audience members to be polite and be respectful while in Black Immuno space. If we see anything offensive, your comments will be deleted immediately and you'll be removed from the platform. After the conclusion of the week, recorded videos will be posted on our YouTube channel and transcripts will be made available on our website, blackimmuno.org. So, my name is Sheon Olufemi, and currently I'm serving as a research assistant at Elix Belgian Institute, Nigeria, and I'll be the moderator for our science talk section today, which is focused on emerging pathogens and vaccine development. So we have 5 speakers here to tell us about their work, and they have 15 minutes for their presentation. Then we follow with a 5-minute Q&A. So without further ado, let's get started. So our first presenter today is Professor Daniel O'Connell. Daniel has both wet labs and bioinformatics background. His main research interest relates to the analysis of contemporary high-dimension datasets, that is genomics, transcriptomics, proteomics, to elicit the molecular determinant uh, of vaccines response. Daniel completed his doctoral research at the University of Oxford and is now an associate professor of system vaccinology at Oxford Vaccine Group, University of Oxford. It's so beautiful to have you on the show, Professor Daniel.
Thanks, Sean. Thanks for the introduction. So I'll get started. So today I'm going to talk a little bit about systems vaccinology and approach to understanding immune responses to vaccines. So vaccine responses are heterogeneous. So this picture I'm showing here is just a level of functional antibody against a particular vaccine, in this case, group C meningococcus. And this isn't a log scale, this is antibody levels at 5 months of age. So this is after a couple of doses of this vaccine given in early life. And you can see it's quite a spread of responses. And importantly, some responses do not pass this blue line, which is the putative level of protection. So these are potentially some primary vaccine failures. But you can see there's a big spread of responses here, given that the age group of these individuals is all the same and they receive the same vaccine. Why are vaccine responses so varied? And this can become a problem because waning immunity can result in rapid loss of this protection. So you can see here in this particular plot, although most individuals have adequate antibody levels at this 5-month period I previously described, by 12 months of age, this can wane rapidly, so that around 50% of children have now antibody levels below the putative level of protection by a year of age. And for another encapsulated bacteria, in this case Haemophilus influenza B, more than 25% of children now have antibody levels below this putative level of protection. So it's a variable response that can wane rapidly. And this can be particularly important as specific antibody is essential to maintain continuity of vaccine-induced protection. Some of these bacteria can rapidly invade. So this is, for example, these encapsulated bacteria such as group C meningococcus can rapidly invade. So after about 3 days of acquiring these bacteria, they can invade and cause disease. So you think about this particular plot is showing a memory response. So after encountering another exposure to these antigens, you produce antibody levels again, but this can be a bit too slow to protect individuals. So we really need to maintain high antibody levels to ensure protection. So what I've been trying to do is use various types of approaches, and particularly these omic approaches, to understand the variability in vaccine responses. So I'm going to talk about a bit about the genetic analysis I've done and a bit about transcriptomic analysis I've done. Essentially, this is to look at these factors and how they impact vaccine outcomes. So our antibody levels, as well as whether we're protected against following vaccination. So why look at genetics? Well, for a few years now, a few decades, we've known that vaccine responses are highly heritable. So if you take identical twins, they have very similar vaccine responses compared with even dizygotic twins that share only 50% of genetics. So this shows that a lot of our responsiveness is determined by our genetic makeup. So I've just highlighted a couple here for tetanus, vaccine responses, and hemophilia influenza B vaccine responses that are highly heritable here. So heritability means essentially how much of the variability in this phenotype, in this case antibody responses, is due to our genetic makeup. So I just want to talk a little bit about a recent study we published last year to look at a genome-wide association study of COVID-19 vaccine responses. So we had some participants enrolled in our Oxford ChAdOx vaccine trial. So we separated this population into an initial discovery cohort to look for genetics underlying vaccine response, and we had a subsequent set of individuals who we aimed to replicate our findings. And what we found in the discovery cohort was an association between particular genetic single nucleotide polymorphisms, and in particular, you can see on this Manhattan plot. So just to, if you're not familiar with Manhattan plots, these are single nucleotide polymorphisms across the chromosomes of a human, and each particular dot is a single nucleotide polymorphism starting at the beginning of chromosome 1 and finishing at the end of chromosome 22. And you can see this peak is just an association peak. So this is a minus log 10 p-value, showing that there's a statistical association in this region on chromosome 6. And this region of chromosome 6 is where the major histocompatibility complex genes lie. And you can see particularly here the particular HLAs that are associated with vaccine response. So in the discovery cohort, this initial nearly 1,200 individuals, you had this association peak highlighting our HLA or our MHC that was associated with the level of spike antibody following COVID vaccine. We were then able to replicate that in a replication cohort, and again, just showing that the presence of particular HLA classical alleles were associated with the level of antibody post-vaccination. And this was published, as I said, last year, so you can look into a few more details if you have the time. Not only did we see association between antibody levels to COVID-19 vaccine, but also this same HLA alleles were associated whether you were going to have a breakthrough infection. So this plot here shows just the breakthrough infection incidence, and you can see if you have this particular HLA allele, you have a lower probability of having a breakthrough infection. So you have higher antibody levels, and you also have a low probability of having a breakthrough COVID-19 infection. So this was adjusted, as shown exactly what we adjusted for in this analysis, but that shows the findings. We also have suggested in this paper that the association is actually causal. So if you do some analysis here to look at the structure of this particular classical HLA allele, because we predicted that this particular allele, this HLA DQB10602, has a different property in binding to SARS-CoV-2 spike protein. And as I said, the further details are described in the paper. This has subsequently been independently validated by other groups. So this is using data from the UK Biobank. So this is over 180,000 individuals that they analysed in this study. And it's now, there's a preprint available you can look in the full details of this manuscript, but you can see there's a very clear association here in this particular region, MHC/HLA. And again, when you look at this particular HLA classical allele, you can see that carriers of the allele have higher antibody levels. And in this particular study, they didn't have such a quantitative measure of antibody, but rather just a presence or absence of antibody against spike protein following vaccination. This study went on then to use an approach called Mendelian randomization to suggest that antibody against spike confers around 50% of protection against severe disease. So it's clear that this sort of genetic analysis can show and explain some of the variability in vaccine responses, as well as our variability in protection following vaccination, and also can be used to actually suggest exactly the mechanism of protection using this Mendelian randomization. Don't really have time to describe what Mendelian randomization means, but essentially it's the genetics underlying the correlation between the genetics underlying the antibody response and the genetics underlying the risk of a breakthrough infection. So to summarize this initial part that I want to talk about today, immune responses to vaccines are heritable. We showed that that HLA was reproducibly associated with our response to anti-S following COVID-19 vaccine and also the protection following this vaccination. The next bit I want to talk about in this presentation is the transcriptome. So I want to talk about a study which we published a couple of years ago now to try to use an omic approach to elucidate the determinants of a particular vaccine immunogenicity and reactogenicity. So a little bit of background about this vaccine. So Suliman B vaccine is an efficacious vaccine against capsular group B meningococcal infection. So this is just some epidemiology of group B meningococcal invasive infection in the UK. And essentially what it shows is that that group B is the major cause of invasive meningococcal infection in the UK. And since we introduced this vaccine, we can see a reduction. So this is the incidence that we saw post-vaccination implementation. And this line is where we had predicted the cases to be. So you can see this is the efficacy of the vaccine. And you can see in this control group who didn't receive the vaccine, there's no change in the epidemiology, the incidence of this disease. So this vaccine does work, but it's quite reactogenic. So more than 50% of individuals, if you highlight in this middle bar here after the second dose, so if you have 2 doses of this Suliman B vaccine along with routine immunizations, about 60% of these infants develop a fever greater than 38. So in the UK, this has resulted in the use of prophylactic paracetamol in order to deal with this increased rate of fever. So fever is sort of benign and not that important. However, if you think about the consequences of having febrile events early in life, they do have actual consequences. So you can see here, this is just showing the impact since this vaccine was introduced in the UK. So we have increase in adverse events following immunizations at 2 and 4 months of age, is when this vaccine is given. And the consequence of that has been an increase in admissions, in ward admissions, and unnecessary procedures such as lumbar punctures, and increased use of antibiotics. This is because the threshold of concern for potential bacterial infection in early life is very low. So if you have acute febrile illness in a baby, there is concern this could be a bacterial infection. So this increased rate of febrile events following vaccination has a consequence. And if we extrapolate from some data that was generated in Scotland, we think that about 1,400 extra hospital visits in the UK are resulting from the use of this vaccine. So the vaccine works, but it's resulting in increased rates of fever, which is resulting in extra hospital visits and cost to the healthcare system. So we conducted a study to try to work out what the molecular determinants of this increased reactogenicity was, and also if we could understand the determinants of immunogenicity. So we took a study, we had just over 118 babies, and we took blood samples 4, 24, 3 days, or 7 days after immunization, and did some proteomic and transcriptomic analysis. And the type of analysis we wanted to do is to see if we could understand if there were any markers of reactogenicity and immunogenicity, as well as to see if we could predict who would develop a fever and how well an individual will respond to a vaccine based upon factors in their blood. So this is just a snapshot of the high-level information of the blood transcriptome analysis. You can see a peak in differential gene expression 24 hours after vaccination. So these are the different time points: 4, 24, 3 days, and 7 days. And you can see, if you're not familiar with volcano plots, the y-axis is the significance level and the x-axis is the degree of fold change. So upregulated in red and downregulated in blue. The significant ones are in blue or red. And you can see that there's a real perturbation of gene expression 24 hours after vaccination. And when you do principal component analysis, which essentially is to look for the structure of the transcriptomic data, you can see that the 24-hour samples really cluster separately from the other samples. And you can actually try to work out what genes are driving this. And CD177, which is a marker of neutrophils, really came out as a strong determinant of the structure of these data. So the reason for this is because we see an increase in neutrophils 24 hours after vaccination. So we've got here in purple the individuals who had the 4C B vaccine plus routine immunizations, and in green we have the control immunizations alone. And 24 hours, you can see here, you can see an increase in neutrophil count. Even when we corrected for neutrophil count, we still saw differences in gene expression, but a lot of this gene perturbation was attributable to genes related to neutrophils. So another thing we did, as I mentioned, we looked at baseline gene expression profiles to see if we could predict immune responses. So these two plots here, first on the left-hand side, we wanted to predict antibody levels following immunization. And what we did is we separated the data into a training and validation set. So we built using machine learning a model to predict antibody levels. And you can see it here. It does very well in the training data set. Maybe not too surprising because these are data used to train the model, but more importantly, in the validation data set, you can again see that using the same model, and it hasn't seen these data before, using the transcriptome, we could predict the antibody level post-vaccination. Similarly, for the reactogenicity data, predicting whether they have a fever or not, we could in black we have the training model, area under the curve. And in red, we show that using that same model in an independent data set, we could again predict those who develop a fever. So essentially, to summarize this, we show distinct gene expression profiles after this vaccine, which has an interesting reactogenicity profile. A lot of this gene perturbation was dominated by neutrophils, and that machine learning could enable prediction of immunogenicity and reactogenicity using pre-vaccination gene expression data set. So the final study I want to talk about is to use molecular correlates of protection against the typhoid fever vaccines. So this is the experimental design. In this experimental design, we gave 2 typhoid vaccines to individuals enrolled in a study, and then we challenged them with Salmonella typhi, and then took blood samples at various time points. A little bit about the epidemiology of typhoid fever. So it's a systemic illness caused by Salmonella enterica cerevar typhi, and it causes between 12 and 20 million cases per year and over 150,000 deaths. It's mainly seen in low and middle-income country settings due to poor sanitation and contaminated water and food. So vaccines have recently been developed, or vaccines have been developed for some time against typhoid, but new vaccines have been generated really recently. But vaccine-induced immunological protection is not fully understood. So in this study, we wanted to see if we could use, again, an omic approach to understand the immunological determinants of protection following vaccination. So as shown in the previous slide, we had 2 vaccines. We had a plain polysaccharide vaccine and we had a conjugate vaccine. And this is, again, a principal component analysis looking at various time points of the gene expression data taken from blood. And just to highlight that the main changes in gene expression we saw were at typhoid diagnosis. But we also saw quite a change in differential gene expression post-vaccination. So before they were challenged with typhoid, we were able to see 7 days after vaccination differences in gene expression. And interestingly enough, we saw, you may not be able to see it clearly here, but we saw lots of immunoglobulin gene segments which are upregulated following vaccination. So what we aim to do is then to take these genes that are upregulated in the immunoglobulin genes, and then to see specifically what these might bind to. So immunoglobulin genes form the B cell receptor. So we extracted B cell receptor sequences from the bulk RNA sequencing data. And this was recently published just earlier this year, so the reference is found on the previous slide. But what we could see at a high level was a hyper expansion of clonotypes in individuals who were vaccinated but were protected following challenge with Suliman typhoid, compared with those who were challenged and developed typhoid disease. And we saw a significant clonal expansion in these individuals. You can see here, so this is a Gini index of a measure of diversity. So you can see it's increased clonal expansion in the individuals who had the conjugate vaccine, who were protected following vaccination compared with those who developed disease. And when we directly compared those who developed disease to those who didn't develop disease, we saw an increase in this particular V gene usage, so a particular variable gene in immunoglobulin, which is V323. To further repertoire analysis, we saw increase in particular clusters. So there was an increase in clustering in general in those who received the conjugate vaccine and were protected following challenge. You can see here it's just an increase in clusters shown 7 days post-vaccination in the recipients of the conjugate vaccine who are protected, so non-typhoid diagnosed, compared with those who developed typhoid. And in particular, we found one particular cluster which was very abundant in the conjugate recipients who were protected, and it was also seen in the protected individuals who received the polysaccharide, but to a lesser extent, and was much less abundant in those who developed typhoid. And this is just showing again some of these data, the counts of this particular cluster of sequences in the protected individuals compared with those who developed disease, in both the conjugate group and the polysaccharide group. This is the particular motif and the conserved parts of the variable, the CDRH3 that we identified. And when we looked at these sequences and compared them to experimentally defined known binders to the components of this vaccine, we could see that 7 days after vaccination, we identified particular sequences that are very close to known binders, including sequences that were identical to the known binders. So essentially what this is showing is in this cluster 184 included sequences that bind to the vaccine component. So it's a specific response, or it seems to be a specific response to vaccine component that was associated with protection. So finally now, just to summarize some of the data I've shown, these studies show that the utility of omic approaches in elucidating novel immunobiology, which can hopefully feed back to future vaccine development implementation. We reveal genetic factors underlying heritability of vaccine responses, and demonstrated the ability to predict vaccine responses using some of these high dimensional data. We could predict vaccine immunogenicity and reactogenicity. We also potentially highlighted a novel vaccine correlate protection. So here, just my acknowledgement. So it's quite a large group that I'm based in, the Oxford Vaccine Group, and I've highlighted particular key individuals here, as well as some external collaborators. Thank you.
Yeah, thank you very much, Professor Daniel. It's such a really really great science, having to talk about the genomics of immune responses all across different populations, genome-wide association studies and the likes. So, do we have any question? Question from Professor Daniel as regarding his works, which is all around the immune responses, trying to get. So Daniel, I am kind of inquisitive about your work. The very first one is when you talk about genome vaccine vaccine. Was the study specific for a type of COVID-19 vaccine, or you just do multiple COVID-19 vaccines and try to see the associations with the genetic variations and others?
That's a good question. So you're breaking up a little bit, so I'll repeat the question. The question was whether we saw the genetic association for one particular COVID-19 vaccine or for multiple COVID-19 vaccines. So we started in the discovery phase. We looked for association between the ChAdOx1 COVID-19 vaccine, which was developed in Oxford and was commercialized by AstraZeneca. But we were able to replicate that for the Pfizer-BioNTech vaccine. And other groups have subsequently replicated that these associations for other COVID-19 vaccines. So the other group that I mentioned from the UK Biobank study, they looked at all COVID-19 vaccines that were used in the UK. So that would include Moderna as well.
Thank you very much for that clarification that you had to use Visa and the likes. Okay, then another question that I have. Sorry if my question is getting too much. Another question that I have is when you try to use omics approach, particularly the transcriptomics approach, in trying to measure cellular heterogeneity for vaccines that has to do with 4-HEM-EBV. I'm so sorry if I couldn't get the name so well. The question is pre-vaccination and post-vaccination are the neutrophil linked genes that you identified to be significantly differentially expressed. Are they the same, or you have variations across these two pre and post vaccination rather?
So what we see in that particular study for this 4-HEM-B vaccine, we see an increase in lots of neutrophil-related genes, but there's also an increase in neutrophil count in general. So there's often a question related to whether you see just an increase. Some of the findings you see are just an increase in cell abundance or the change in the cells are expressing. When we corrected for neutrophil number, we saw large changes in differential gene expression. So I think it's both of those things. You see an increase in neutrophils and a change in the transcriptome of those neutrophils. But single cell type analysis is really needed to more definitively answer your question.
Okay. Do we have any other question?
I can see one came up actually from Yasin. It comes up on my screen, which was just a question related to, is vaccine heritability associated with epigenetic changes in the genome? It is. We published a paper. I didn't include it because I just want to include 3 studies, but we have done some epigenetic work looking at DNA methylation. And the take-home message from our work so far in epigenetics is that your epigenome changes a lot early in life. I work in pediatrics, so a lot of the vaccines we were looking at are given to a pediatric population, and your epigenome is changing a lot early in life. And we also link some of those changes to how well individuals respond to vaccine. So the question about how epigenetic factors can be heritable is a sort of different question, which is outside of my area of expertise. But we definitely see epigenetic factors associated with vaccine responses as well as genetic factors.
Yeah. Thank you so much, Yasin, for the question. Let's see if we can take one more question before we move to our next speaker. Okay. I don't think we have any other question. So, in the absence of no other question, thank you so much, Professor Daniel. It's nice having a great talk to you. It's nice having a great science, and we do hope that you keep moving the science banner forward. Thank you so much.
Thank you.
So, our next speaker for this section is Dr. Sara Suliman. I don't think if I get that right. So, Dr. Sara Suliman is an infectious disease immunologist and an assistant professor in the Division of Experimental Medicine at the University of California, San Francisco. She has an extensive experience in biomarker discovery and clinical trials of TB vaccines. Our lab uses multiple approaches to understand risks of and progression of active tuberculosis disease and treatment response, and to identify candidate TB risk pathways and their role in TB pathogenesis. Previously, at the Brigham and Women's Hospital, she was also the lead of the Mass General Brigham Diagnostics Accelerator Lab to evaluate the accuracies of new COVID-19 diagnosis. Thank you so much, Professor Sarah. It's so nice having you on board.
Thank you so much, Shion. So I'll just put this on presenter view. So thank you, first of all, for this fantastic forum, and congratulations to Black in Immuno Week, Black in Immunology again for a fantastic symposium. So what I wanted to focus on today is lessons in pandemic preparedness with a focus on diagnostics, especially that we want to have the tools to detect emerging pathogens. But there are lots of considerations in having proper diagnostics that are evaluated properly before deploying them for mass screening. So I think we've all become experts in COVID-19 diagnostics. We're familiar with three different categories of tests. One is serological, which were actually popular at the beginning of the pandemic. And these are meant to detect antibody responses against SARS-CoV-2. You can either detect the spike region, which would actually cross-react with vaccine-induced responses, or nucleocapsid, which would differentiate infection from vaccination, unless of course you are vaccinated with whole inactivated viruses. And then molecular assays, so those are PCR tests, and we can either use it as a binary test of whether or not SARS-CoV-2 is present, and also we can use it as a quantitative measure of the viral load by looking at the cycle threshold. And then the third category, which we're becoming a lot more familiar with now, are rapid antigen tests, which are meant as point-of-care testing that the end user can use at home. And these are also very convenient for global implementation in resource-limited settings where you don't have access, for example, for electricity for PCR testing. So why did we need evaluations for COVID-19 tests? So there were two seminal stories that came out at the beginning of the pandemic where a lot of tests have been recalled after being deployed because it was clear that they were not accurate in detecting SARS-CoV-2 either by serology or by PCR. In response to that, the Mass General and Brigham hospitals formed a COVID Innovation Center. And within the COVID Innovation Center, there were four pillars. So the pillars for pandemic response included devices with a specific focus on PPEs because we were having a lot of supply chain issues at the beginning of the pandemic, as we all recall. Diagnostics, data analytics, which would facilitate early data sharing between investigators across the ecosystem, and then therapeutics. And of course, there were already existing efforts across the country for vaccination, but when we think about a framework for pandemic preparedness, we also have to think about accelerating vaccine development. So that's an important pillar to have if we want to create a framework for pandemic responses against emerging pathogens. So because of the shutdown that happened early on in the pandemic, about 1,000 volunteers across the ecosystem, all the way from undergrads and technicians to full professors, and contributed to this effort. And it really highlights the power of pulling intellectual resources together for participating. So the pillar that I've been mostly involved with was the diagnostics pillar, and this is because I had already some background in biomarker development for TB. So the mission of the diagnostic pillar of the center was to identify, assess, validate, and implement accessible and scalable diagnostic solutions in response to COVID-19. And we wanted to marry the different use cases for different diagnostics and appropriate technologies that would match those use cases. For example, for mass community screening and assessment of transmission, you need different types of diagnostics than what you would need for clinical assessment of COVID-19 patients. As part of the diagnostic pillar, we had an accelerator lab, and this was actually my first run at running a lab, and it was very exciting and actually motivated me to start properly applying for faculty positions after, because I really enjoyed working with a team of young investigators that were very energetic and fantastic. So Sally as Marlene, Zach, and Sujata. David Walt was the head of the diagnostic pillar. Lauren Ritterhouse was a pathologist who actually started running the lab and then had to leave for maternity leave. And then I took over running the lab. And then we have two fantastic lab managers, Bianca and Savannah. So when we think about diagnostics, we have to think about the framework for measuring the accuracy of diagnostics. And I know this is not your usual immunology analysis. So just very quickly, what you do is look for, plot your data in this 2x2 table, which we call a confusion matrix. You have to assign a gold standard, which is also part of the problem, because you need some measure of the truth, whether or not you have the disease of interest. So in this case, we bench our diagnostics against PCR. And remember, PCR tests were just starting to emerge, so we did not have enough of a track record to actually ensure that they were giving us accurate results. But this was the best that we can use to benchmark our data to begin with. So then you stratify your results by the gold standard into positive and negative. And then your new test, which you want to validate, would be the predicted class. And then you stratify that into positive and negative. If both agree to be positive, that's your true positive. If both agree to be negative, that's your true negative. And then if there's a disagreement, you can have either a true positive or a true negative. And then the measurements of accuracy include sensitivity, which is how many of the cases are detected accurately. And specificity, which are how many of the uninfected controls or the controls are detected accurately as well. So people who are uninfected, are we able to call them correctly by the test? We can also flip the denominator. So then you can measure precision, which is the positive predictive value. So how many of the tests that came out positive actually correspond to a true result. And similarly, we can measure negative predictive values. And in our lab, we also measured interoperator reproducibility. So if two different operators run the same test, are they coming up with the same result? And then the limit of detection. So how low of either a viral load or antibody response can we detect using the different assays that we have. So why do we need serological tests? These, as I mentioned, were popular at the beginning of the pandemic. And the thought was to measure the extent of transmission in the community. And seroprevalence studies are still widely used to estimate the prevalence of different infections across different classes, viral, bacterial infections, etc. If you want to dynamically measure the transmission of SARS-CoV-2 or any other viral pathogen, ideally when you identify an index case and you know that that person has been in contact with a number of uninfected people, then you can later on use serological tests to determine the extent of transmission. So you can have people who are asymptomatic or people who are symptomatic, but then both the symptomatic and asymptomatic cases would turn out positive on a serological test. And there was a lot of discussion at the beginning of the pandemic about using serological tests as immunity passports. And of course, that comes with a lot of scientific and ethical issues. So for example, ensuring that someone has antibody responses against SARS-CoV-2 to allow them to go on planes, for example, or allow them to come back to work. Of course, vaccination kind of mitigated this concern because you don't have to rely on naturally induced immunity from infection. But still, there are very clear ethical issues with immunity passports and scientific issues because we can't accurately determine whether you just have a negative test because you were not exposed or because the limit of detection for the test did not allow you to detect antibodies in that sample. So we published this paper where we benchmarked the performance of 20 different serological tests that were coming up at the beginning of the pandemic. Some of them had emergency use authorization and some of them did not. The goal of serological tests, of course, is that they would be decentralized, so someone can just buy them off Amazon or any other supplier and then use them at home. So you need to have specific criteria to ensure that that's fit for use. And the goals of direct-to-consumer tests include that you use accessible samples. You don't want to go home and, have a bronchoalveolar lavage collection or, require a blood draw, for example, at home. Ideally, you want to use accessible samples like saliva, finger prick, or urine tests. And then it should be easy to perform by non-healthcare providers. So you and me who've never gone to medical school should also be able to perform these tests. They should ideally have a rapid turnaround time. More than 15 minutes, you start to question whether this is a reliable assay or not. And the interpretation should be easy. So the lateral flow assays are the form that we're most familiar with, and it's just a matter of whether or not there's a line. So then that would tell you whether or not you have a positive serological test. So we used samples that were biobanked, and at this point, the controls had to be pre-pandemic samples, so samples that were collected before December 2019. And there was actually some discussion if we needed to go even further back because the actual timeline of when SARS-CoV-2 started to become transmissible in the US is still not clear. But at the time, December 1st was generally a good estimate. And then we had negative samples that were pre-pandemic and positive samples that were confirmed using one of two RT-PCR platforms, the Cepheid or the Panther. And within the SARS-CoV-2 negative samples, we also included HIV positive samples. And the thinking behind this is that we know that there's evidence of generalized immune activation when someone is infected with HIV. So we might have false positive results just as a result of the HIV infection. So we included those in the analysis as well. And then we looked for sensitivity, specificity, interoperator reproducibility. And these were conducted in a lab, which is not how you would conduct the tests at home, of course, but this gives us an initial benchmark as to whether or not the accuracy of the tests were sufficient for field validation. So we sourced these 20 lateral flow assays, and they passed initial screening criteria. If you want more details, you can follow them on this link. And as we know, antibodies come in multiple isotypes. So IgM will tell you if there's a recent infection, and IgG will tell you if there's an established or a more remote infection than a few weeks. So if the control line is positive, that means your assay is valid. And if the test line is positive, that confirms that there's evidence of antibodies in the sample. So I'm showing you a heat map that summarizes a ton of work by different volunteers that worked in the center. And in each row, you have one of the assays that we evaluated, and each column is one of the samples. So you can easily see right away that overall the negative samples had negative results, but there was some evidence of false positive results, which could actually be due to cross-reactivity, for example, with other coronaviruses other than SARS-CoV-2 that people just get infected with seasonally. And then you can see in the COVID positive region, whether you look at IgG or IgM, that there were samples that were consistently coming up as negative. So those could be people that were infected but did not mount a sufficient immune response. And then you can see, if you look horizontally, that some of the assays were just always bad and did not give us accurate results. So of course, that takes a very clear hit on the sensitivity of the assay. So this gave us a very quick snapshot on the performance of the different LFAs that we sourced. And ideally, you want to translate the performance of these assays to a positive predictive value. So once you deploy them in the field, where the prevalence is very variable. So we had seasons where there was a lot of transmission and seasons where there wasn't a lot of transmission. So we wanted to take into account the accuracy of the test as a function of the prevalence in the community. And we know that accuracy increases the more prevalent the disease is. So if you have a lot of transmission in the community and a test comes out positive, it's more likely that this is an accurate result. However, if you have lower transmission in the community and the test comes out positive, you have to question whether or not that test result is accurate based on the understanding of transmission in the community. And there's a formula to address that. Bob Kitchen actually made this app, which I just checked before this presentation was still active. So if you are interested, you can either manually input your sensitivity and specificity of a given result based on the information brochure that comes with these diagnostics. And then based on your understanding of the transmission in the community. So, I mean, this is made for COVID-19, but you can actually apply it for any disease where you know the prevalence. Then you can estimate what the positive predictive value would be based on the sensitivity, specificity, and the prevalence in the community. So it's a very useful tool, and it allows the user to interpret what the result means, because a positive result is only positive in the context of the accuracy of the test and the dynamics in the community. So moving on from serological tests, we also wanted to test different point of care tests like antigen tests and molecular tests. And we had a point of care working group within the diagnostic pillar that actually scanned hundreds of different available diagnostics that came out in the market. And remember, a lot of companies repurposed their pipelines to start working on COVID-19. Some of them did not even have a track record of working on COVID-19. For example, there were hair companies that started making point-of-care tests for SARS-CoV-2. We wanted to apply a certain number of criteria, including accuracy, the reputation of the company, supply chain, throughput and turnaround time. We wanted to prioritize tests that would give us, that can analyze a lot of samples in a short amount of time, and then the use case. So we down selected that to a number of different assays, and we were actually able to contact a few of these companies and source their instruments so we can conduct an evaluation. And this was also a helpful partnership with the developers, because they were interested in having real life labs validate the performance of their assays when they didn't have access to clinical samples. So the two companies that we worked with were Fluxergy and OnSite Gene. Both of them, unfortunately, did not get emergency use authorization because the performance of the assays was less than ideal. It was 70% for Fluxergy and 50% for OnSite Gene for the sensitivity. And we still relied on the Crimson Biobank at the Brigham. And this is just a lesson that keeps coming up, which is the utility of having biobanks to centralize the evaluation of new tests, which just means in future pandemics, we have to think about having the infrastructure for biobanking samples quickly and making those available for investigators. We had some issues because initially in the pandemic, the charts were not very clear, did not include CT values from the original tests that were conducted, but we managed to still get at least positive and negative calls from the samples that we were able to validate. One little story that I wanted to highlight is that we were very interested in this platform called Acula from Misa Biotech, which was a handheld PCR device where you can get a result within 30 minutes. and it already received emergency use authorization, so it was already FDA approved for use in the hospital. And we were specifically thinking about it for maternity wards, where you can triage moms that are coming for delivery into either COVID positive areas or COVID negative areas. We built a partnership with Misa Biotech. They were close to sending us the instruments, but then told us at a meeting right before that the NBA bought the entire supply chain from November 2020 to February 2021. So then this became less on our list of priorities. And this also highlights a lot of the inequalities and issues that come up with access to diagnostics during the pandemic. And another problem that, of course, we had to address is the standardization of different tests across different platforms. So we want to make sure that we have a way of making sure that if you test someone in Dakar, in Senegal, for example, and take the same sample and test it in San Francisco, that you would get exactly the same result, or at least a similar assessment of the viral load. But to do this, you need to have standards that can benchmark the performance of your assays to a standardized unit or the international unit, which is what the WHO does for different diagnostic tests. So we had this amazing partnership across 14 different labs, and we sourced 7 widely available molecular standards and benchmarked the performance, not the performance, sorry, the quantitation of these different standards against a unified standard, which was made by the National Institute for Biological Standards. And the goal of this is to harmonize how we quantify each of these standards, so that when someone uses this standard LGC, for example, to quantitate the viral load in a specific sample, if another lab that uses a different instrument and a different standard, they can also report the viral load in international units. We finished this analysis, and it should be published very soon. Another lesson that came up in this working group is at which point in developing diagnostics do we need standards? So when we develop a testing platform, we need to, first of all, know what the sequence is of the virus, develop primers and probes, verify the accuracy of these primers and probes, and then there are different stages where we have, sample collection. What are the different methods of sample collection? Cold chains for transport, etc. And then the analytical part, where you actually qualify the performance of the assay, and then post-analytical is once you have the data, what does the data actually mean? And you need standards for all of these steps. You need clinical standards, both negative and positive. We need international standards, so you can have unified units for reporting different quantities of the pathogen. And then you also need synthetic standards, so you can quantify the viral loads, the different buffers, and the matrix that you analyze the samples in. And you need to standards for the performance of the labs themselves. So you want to make sure that the labs are able to produce reproducible data. And then once the information is out, you need minimal information standards for how you report that data and how you make it accessible for other people to reanalyze it and reach the same conclusions. Finally, I wanted to talk about rapid antigen tests, because that's the testing platform that we're most familiar with. We partnered with the city of Holyoke in Massachusetts, which was underrepresented in testing in general. This underrepresented community had a large Latinx population. This was a test that was actually developed in Massachusetts, Access Bio. By Access Bio, it's called CareStart, where it's just a rapid antigen test where you need both a control and a test band, and then all the PCRs to benchmark the performance of the test were done at the Broad. And this is the team, which was a fantastic group of people. It was a collaboration between the city of Holyoke, different investigators, and an amazing research team that was actually in the field testing people wearing full PPE. So we did the collections in early 2021, before vaccines were available to the public. So that allowed us also to get a sense of transmission in the area before vaccines were widely used. And we had about 50% sensitivity, and this is actually standard for point of care tests, because you need intact antigen, meaning that you have intact virus. So the PCRs tend to overestimate the prevalence of SARS-CoV-2, because you can still be PCR positive for a long period of time because of residual dead RNA in the nasal swabs. So the tests are still being used, and I believe they received emergency use authorization. But more importantly, we also built this global implementation group where we wanted to facilitate access to different rapid antigen tests to collaborators in different countries. So we worked with PIs in Kenya, Bangladesh, Malawi, and we partnered with BD to evaluate the BD Veritor. And BD was the company of choice because it's known internationally, and people felt comfortable with the reputation of the company for using their tests. And one of the main rules of the global implementation group, at least from my side, was that there would be no helicopter science. We would have true partnerships with the labs that we were collaborating with. And this was a recurrent theme from the collaborators in the different countries. So this is the BD Veritor. You can either read the results by the naked eye, or you can read it through this BD Veritor Plus reader. And for the most part, there was high concordance between the two results. But in cases where the viral load was low, we noticed that there was a discordance where we can only get the result with using the reader. And the studies were led by the investigators in the different countries. Jannat in Bangladesh wrote this paper where she showed that the performance of the BD Veritor was superior to the performance of other antigen tests that were widely used in Bangladesh at the time. Standard Q was one of these antigen tests. And the other study was performed in Nairobi, and you can see that they also reached similar conclusions about the sensitivity of the BD Veritor. And of course, as we know, the sensitivity is always higher in symptomatic individuals than asymptomatic individuals. And in Kenya, actually, their lab was proficient in implementing different types of point-of-care diagnostics. So they also conducted interviews with both the healthcare workers as well as the clients or the patients, the people who actually came to use these assays. And I remember at the time, nasopharyngeal swabs were the standard. So a lot of people were using them for testing outside of the US. So it was refreshing to them to use anterior nares swabs instead of nasopharyngeal swabs. And that came up in the interview results. So just to conclude, the standardized evaluations of diagnostics for COVID-19 or any emerging pathogens are very critical. It should be tailored to the use case, and the community of scientists that came together accelerated the progress, and we hope that this infrastructure can now be deployed to other pathogens. And it has definitely built a model for infectious disease preparedness, both locally and globally. And new questions, of course, that came up after this exercise is, using this platform also to evaluate breakthrough infections and the performance of these diagnostics against different variants of SARS-CoV-2. And just to end, I'm from Sudan, and I want to highlight that in Black and Immuno Week. And Sudan has been, unfortunately, in a war since April 2023. At least 10,000 people have been killed, and more than 7 million people have been displaced. And conflict zones are breeding grounds for outbreaks and also new epidemics, because there's a lot of interruption to antibiotic use, which would be a breeding ground for antimicrobial resistance. And this is something to keep an eye on, especially when access of healthcare professionals to these regions is compromised because of the instability in the region. And pandemic surveillance efforts should not just focus on new and trendy pathogens that are just emerging. We have to think about endemic diseases that still have inadequate testing, like TB, which is the leading infectious disease killer globally. And after 2 years of COVID-19, actually TB went back to surpass COVID-19 in the annual mortality. So the attention definitely needs to be refocused into existing global health problems and not wait for emerging pathogens to focus our efforts. And with that, I would like to acknowledge the groups that participated in contributing to the COVID-19 Innovation Center. And this is my lab at UCSF. And if anyone here is applying to graduate programs, I highly encourage you to apply to the biomedical science program at UCSF. The deadline is coming very soon. So add that to your list. Thank you. All right. Shaun, you're on mute. I don't know. Are there any questions? I see Yassine is saying thank you for highlighting the war. Thank you, Yassine, for listening. Are there any other questions? Should we move on to the next speaker, Shion? Oh, no, Joel has a question. Can you tell us more about how your lab tackles these global problems? Thank you, Joel. So I rely very heavily on international collaborations, my lab focuses on TB. I think we had our run with COVID, although we're still interested in COVID and how SARS-CoV-2 infection can contribute to TB reactivation. So we have partnerships with a group in Lima in Peru and the same group in Nairobi, actually, that contributed to the COVID-19 diagnostic study. I think building true partnerships and true horizontal relationships between, the labs in the US and labs in LMICs, where we're not just, helicoptering and collecting samples, but actually having regular discussions, contributing to the science together. And now, for example, I'm hosting one of the trainees from the lab in Peru in the lab right now to learn immunology techniques. I think it's important that you build trust and come up with the scientific questions together, because there are things that we think we know, but we don't, I think the network of these collaborations is the basis for how we tackle these problems. And I think that also informs what questions are relevant. Just keeping that in mind. So in terms of techniques that we use in the lab, we also rely on omics approaches to come up with hypotheses about what host factors are involved in mediating susceptibility either to TB disease once you're infected or to response to treatment once someone is diagnosed with TB. So we rely on different, metabolomics approaches like mass spec, immunology, RNA sequencing, single cell RNA sequencing, and then we come up with hypotheses that we back translate in the lab into testable models like in vitro approaches using CRISPR or mouse models. And then Medina has a question. When it comes to continuing work on endemic pathogens, what do you think can be done to maintain a focus on these? I would say the most successful pathogen or community of virologists has been the HIV community, and there has been a lot of advocacy and a lot of grassroots community organizing around these issues. The advocacy in TB is building up, but it's definitely nowhere near what we saw in HIV. And actually, the incidence in TB, ironically, increased after COVID, except in people who are also infected with HIV because of how strong the HIV programs are. So activism is really important to maintain the focus on these. And getting the attention of funders, I've learned that a lot of philanthropists, fund things because they're personally affected by different infections. For example, one of the foundations that funds long COVID is because one of the philanthropists is also affected by long COVID. I'm definitely not advocating that people get the disease for them to become philanthropists and contribute to funding, but it definitely helps when those stories become more personal to people who are in positions of power, to either provide resources or funding, or create programs that can facilitate progress in these areas. Right.
Yeah, thank you very much, Dr. Sarah, and thank you for such mind-blowing research. It is really, really interesting. Because trying to look at Africa, that's one of the major issues that Africa has gotten when it comes to COVID-19 testing, which has actually made Africa to be kind of underrepresented, under talked about when it comes to the detection of this thing. So one major question. You talked about Aquila, which is an interesting platform,
Mm-hmm.
which we suppose that we get the better light. So all those Aquila kind of manage, let's say, false positivity, because one major issue that we have with rapid kit testing is false positivity, false negativity, and this is actually increasing the number of COVID-19 testing, that it is increasing proportionately, which might not actually depict the true picture. So what does Aquila, which happens to be a kind of a modular RT-PCR testing, which happens to be the gold standard, how does it kind of help reduce this false positivity or false negativity testing? Thank you.
Yeah. No, this is. I think I'm echoing now. Shun, maybe just put yourself on mute. Okay, yeah. So I think it's interesting because you call it false positive testing, but it's a result that actually tells you something. Of course, you want to clarify using negative controls that the assay, the buffers are not contaminated, the primers are not contaminated, etc. But the problem with SARS-CoV-2 is that the RNA fragments linger in your nasal cavity for a really long period of time. So even though you don't have an active infection, you don't have intact antigens, 1 month after infection, you can still come up positive on a PCR platform. So I think the evaluations should definitely account for that, and they should account for having controls against related coronaviruses as well. So you want to have specificity controls, so things that could potentially cross-react with the primers that you have, or if it's an antibody or antigen test, cross-react with the assay that you have, to ensure that it is truly specific for the virus that you're interested in. So initially, the emergency use authorization guidelines were very minimal and actually really scary. Like the EUA approvals that some of the platforms got were based on 20 positive and 20 negative samples. Initially, it didn't even require clinical samples. Like, it was enough to validate the platforms on synthetic viruses and standards that you can purchase. But with time, of course, there was a lot of competition, and then because of the recall and the inaccuracies that were reported in the field, there was a lot more emphasis on having these controls that ensure that you minimize false positive and negative results. So other coronaviruses, negative and positive controls, yeah, and testing in multiple areas, samples using different viral loads, etc.
Yeah, thank you very much. Thank you for that. Okay, I don't know, maybe we could just take one more question. So, talking about these testing of a thing, and trying to kind of make people get easy and affordable testing, because the it stuck for me was the Aquila of a thing, because I'm looking at it and I'm saying that, okay, we can have something which could actually give good testing procedures to people in Africa, a kind of something modular where people can be able to do. Then how do you see it in terms of affordability? That maybe it will not be all these talking about iPut technologies, whereby they're extremely high and not affordable to people in the black, to the blacks or the people in Africa. So how is Aquila bringing this gap? How is it bridging this gap?
Yeah. So,
Yeah, so
sorry, Shun, can you put
sorry,
this?
Shane, can you put it?
Yeah, so Akila actually highlighted the inequities in COVID testing. It was very clear that they had no, I mean, not just them, so I shouldn't just single them out. There was a lot more interest in profit than there was interest in accessibility to the public. And this is the problem with the private model that we have, where a lot of people saw COVID as a business opportunity, right? So it was very clear that the business opportunity that presented itself with the NBA buying this platform so they can test players was a much more profitable endeavor than making this platform accessible to, I wouldn't call the Brigham Hospital and Mass General resource constrained settings, but even they could not have access. So because there was competition from a more valuable contributor. So that already gives you an insight into the priority that low and middle income countries would have. Um, um, uh, you know, Meeting with these companies, and I sat on a lot of meetings of test developers. It really gives you an insight on the mismatch between the priority of public health and the priority of the biotech companies. And hopefully the new frameworks for emerging pathogens would kind of address that from the get-go, where there should be some restrictions on you know um, um, profitability from a crisis, for God's sake. But you know, I doubt that that world will happen in my lifetime. But I think having the conversation and continuing the conversation and highlighting where we failed in COVID, because we failed massively. The distribution of tests and vaccines and therapeutics was very inequitable, both locally and globally. And continuing to have those conversations and emphasizing where those inequities are would be helpful for future pathogens.
Thank you very, very much, Dr. Sarah. You really, really highlighted some of the core things in the discussion. So thank you so much for joining. In the absence of no other question, or do we still have any other question? So we could just let Dr. Sarah go. Oh, so in the absence of no other questions, so we'll be moving straight to our next speaker, which is Dr. Usman Adamu Dafali. Dr. Adamu Dafali is a senior lecturer at the University of Greenwich, UK, with over decade experience working in clinical setting, research institutions, and academia. He graduated from the University of Nottingham. His PhD research is on the role of bacterial secreted effectors on host pathogenesis and its vaccine potential. Was a multidisciplinary which spanned through the field of chemistry, cell biology, microbiology, and immunology. His current research interests include infection and immunity, cancer, and neurological disorders, and one else that is talking about antibiotics resistance, potential probiotics, and vaccine candidates. He aims to elucidate the contribution of microbial secreted effectors to cancer and neurological disorders and how immune cells subvert the blood-brain barrier. Dr. Dafaliu is passionate about community outreach, youth empowerment, and finds fun in sporting activities, especially table tennis and volleyball. It's good to have you on board, Dr. Usman.
Yeah, thank you very
Yeah,
much,
thank you
man.
very much, man.
I'm not sure if my slides are showing. Hi, Sean.
Slides are on. Please move ahead.
Yeah. Okay. Um, Hello, everyone. Yeah, thank you to the organizers for the opportunity to share my experience. Again, as just going to move a bit away from um, what my colleagues have already done. I mean, showing what they've done in terms of the needed details of the research. And then just to probably share my experience on how I got into vaccine development and specifically one of the pathogens I've worked with. So um, you can see I'm from Atebubu. Atebubu is a town in Ghana known for ATM production. And growing up as a young child in Atebubu, I was growing in a town with an aroma of fufu, which was a local dish made from yam. Again, at that time, we had no light in Atebu, so nights were beautiful with stars and moon playing around. But there was an incident that happened that really stuck with me for a long time was when one of my dear friend, whom we played football together with very promising potential, suddenly died, and that really sent a shockwave to our community. And we were trying to find out what could have caused this sudden death of a promising child. Um, it wasn't clear, but rumors had it that he died out of a heat-related disease, and um, this really still reflect. I still have reflection of my community as we sent to lay him down to rest, and beginning to question myself: could I be the next person, or what could be this that has caused this promising person to die at such an early age? So this really followed me through my thoughts. But as years went by, I proceeded to high school and university in the northern part of the country. So you could see Brunel Half was where I was, and then I went into the northern region. The northern region are considered among the areas to be within the African meningitis belt. So it was here that I identified or I got to know that the heat-related disease that had killed BT was actually cerebrospinal meningitis, and it was it was good to know that these mystery that has haunted me has been released with probably a stark reality or clarity of thought, and that probably made me interested in looking at what causes this cerebral meningitis and what could be done to save the lives of others, such as BT, who unfortunately could not live to meet his full potential. So that led me to joining one of the reference laboratories in Navrongo called Navrongo Health Research Center, where we were trying to look at different vaccines and doing surveillance to see the efficacy of Men A and Men W vaccine. During this period, I was also curious, although I didn't present this research, was just to see that most of these communities are resource poor settings, and as um, uh, Sarah mentioned, that it's difficult to have diagnostics in these facilities, and to be able to diagnose meningitis and administer effective antibiotic treatment, the patient might not have survived. So the practice was, once you suspect, you need to start antibiotic treatment after taking the sample, and then waiting for culturing, which will probably be after 24 hours, to see whether it was actually bacterial meningitis or not. So I wanted to find out common tests that are done in these resource settings, and whether these tests could help us could help us diagnose or screen for bacterial meningitis to ensure effective treatment and to avoid misuse of antibiotics. So a common test was urinalysis. So there's a urine dipstick where you could check for WBC, uh, protein, glucose, etc. And then so we subjected the CSF samples to these, dipping them on these urine dipsticks to see whether there will be any correlation between samples that will test positive for bacterial meningitis and the parameters on the urine dipstick. Interestingly, we saw that there was an elevated protein read on these dipsticks, and then depleted glucose level. And looking at the probably understanding the mechanism of invasion is that these pathogens, in terms of their pathogenicity, might secrete certain proteins to help them colonize, and that probably has resulted in the observed high level of protein in the CSF sample. And then within the environment, they need to feed, and because of that, they probably deplete the glucose within the CSF milieu. So I was interested because at that time the vaccine we're testing was against serogroup W, and we didn't know that the bacteria has the ability to switch its capsules and evade the ability of the vaccine to elicit immune response against the new variant. And there wasn't a vaccine for serogroup B, which was predominantly in Europe, as Daniel mentioned earlier. So I was interested in knowing whether these proteins produced by that I observed its high level could be useful as a marker to for vaccine development. And then my understanding of vaccine was food, and then as food, we need to ask ourselves, why do we need to eat? And we need to eat to protect ourselves. We need to eat to have energy. We need to eat to survive. And with the concept of vaccines, when I entered, I mean, historically, it was the inactivated based vaccine that was used to develop a number of vaccine. Then for the polio-based vaccine, the live attenuated vaccine was developed. And then, so when I was looking at meningitis, the technology that was making rounds was the protein-based vaccine or subunit-based vaccine. So, considering that I saw an elevated level of protein in a CSF sample, I was interested in looking at which of these bacterial proteins could be useful as a vaccine candidate. And that led me to my PhD work in Nottingham, and we were looking at different autotransporter proteins or outer membrane proteins of Neisseria meningitidis to see their potential as vaccine candidates. We did characterize some of those proteins, unfortunately, using the recombinant DNA based technology to overexpress these proteins. Unfortunately, our candidates that we studied were not eventually included in the vaccines that were developed, but our indirect contribution or the role of the proteins we studied on the effect of the other proteins that were used to develop the 4-MeB vaccine that Daniel presented a beautiful data of its efficacy and its action came to light. And therefore, so from 2012 to 2016, when I worked on trying to develop vaccine, eventually, as I said, there was a success with developing vaccine for meningitis, and it has saved life. And I'm sure BT will be happy wherever he is, believing that now these development will probably save a lot of lives of young children to meet their full potentials. Vaccine is important, although we know there has been vaccine hesitant since it's before Christ. Again, we have seen success not just with meningitis, but there has been vaccine success over the years with measles, pertussis, mumps, rubella, and polio. Although we still have some few cases of polio recently in Afghanistan and Pakistan, but hopefully that will be addressed by 2030. One most important concept that we cannot shy away from when it comes to vaccine development, or the game changer, I would say, was the COVID-19 outbreak. And COVID-19 shifted the gear in terms of the process of vaccine development and raised concerns. And some of the concerns that were raised were basically looking at the conventional or the traditional approach of vaccine development would always take 10 to 15 years, looking at the discovery, which is in the lab, the preclinical stage, phase 1, phase 2, phase 3 processes. But because of the insurance package that was provided, it allowed these clinical phases to be run concurrently to facilitate the process. Um, Again, just within April 2020, there were a number of clinical candidates that were already in the various phases of the trial, and one had already received an emergency use. And so I think, as human as we are, we usually have hesitation when it comes to change. So definitely was a game changer to see that, and not that it was a period of time that showed or promoted the use of uh, viral-based vector for vaccine development, because as I mentioned from the beginning, we had the inactivated approach, which we established, was well established, that attenuated based vaccine, the protein-based vaccine, which was what I went into in the field of vaccine development. And you could see that there were a lot of candidates then for developing COVID-19 vaccine. But then there was a new era of ways to develop vaccines during COVID-19, and that involved uh, the viral-based vaccine, which was used again for Ebola, then the mRNA-based vaccine. So these new game changers really probably brought some issues around vaccine hesitance. And I mean, this CDC report shows how people were doubting the safety, doubting the vaccines, whether the efficacy of the vaccines, trusting the government, and they don't believe they need the vaccine, and others probably think others deserve the vaccine better. So all these concerns, as Black in Immuno, it's important for us to be involved in trying to understand our dynamics, to be able to ensure effective communication to better help our community. In the midst of all this, it was a huge global pandemic which really affected the world. But what was surprising was that the prediction that Africa was going to be a disaster tend to surprise us. So interestingly, based on that, we team up with colleagues to review the literature to look at how Africans survived better, and we were looking at COVID-19 in Africa and ovarian victory. And the data from the publication could show that most African countries had low rates per 10,000 compared to America, Asia, and Europe, where there was a high hit. Looking at that, we know that Africa had the highest youth population, and then the female population in Africa was higher than the European and developed countries. And again, we know the ovarian function, which is the estrogen production by females, offers females protection. Literature has shown that it makes females susceptible to cancer, but it also provides them protection to other infections. So based on our review, we saw that estrogen could probably be the game changer, or could be the player in what has resulted in Africa being protected. There were concerns that if definitely it was estrogen, then what of the male population? So we were not sure. We just did a preliminary data work. This is something we would be building on going forward. And what we did were just 2 males who were to be administered with the Johnson & Johnson vaccine. Before that, they were screened to make sure they were negative for COVID. And then once they were administered with the vaccine, their estrogen level was measured before the administration of the vaccine. And 30 days after the vaccine, the sample were taken to analyze the estrogen level, and after 60 days. So we did see that within the 30 days of vaccine administration, there was a spike or there was an increase in their estrogen production. And then after 60 days, there wasn't a significant difference between the pre-vaccine and the 60-day post-vaccine. So we plan to probably continue this on a long term with a larger population to see whether this will be a true picture or not. Again, in terms of the immunoglobulin responses, so for the same group of patients, individuals, I would say, we looked at the pre-vaccine with IgG. There wasn't significant difference between the pre-vaccine 30 days and 60, and that was probably applicable for IgM and IgA as well. So, I mean, looking at vaccines in general, and what COVID-19 has shown in terms of vaccine hesitancy and how Africa was able to survive. Uh, uh, there's a need for us in the minority to start looking at the past, the present, and to look at what the future is like. And for those of us in academia and students alike, to see what the emerging trends are and where we can step in to be able to make the right impact. I mean, data from the COVID-19 does indicate that we've not only improved from the inactivated base viruses vaccine, the viral base, the viral vector based vaccine, the RNA based vaccine. There's an emerging and a protein based subunit based vaccine. There's an emerging area of vaccine development, which is the DNA based vaccine. So for individuals that are interested in this area, they could start exploring the use of DNA for vaccine. And you could see during the COVID period, it received an authorization. An authorization was because it was an emergency, so it could be used. However, we did see that those that were probably the old system of vaccination were the ones that got the most approval, with the protein subunit having 3 FDA approval, the inactivated having 3, the viral having 4, and RNA having 2, which now has probably increased. But the fact that a DNA vaccine got an authorization means this is an area that we could start exploring to see the way forward or the potential of this approach. As I mentioned earlier, I see vaccine development as food, and as members of the Black and ethnic minority group, I think it's safer to say that you would want to eat a homemade food than a food from outside, and therefore we need to work together collectively in trying to be involved in the vaccine development process. We need to and it's been impressive to see Daniel has been involved. And once we all get involved in the space of vaccine development, that would increase trust. That would allow our community to really connect faces and trust to be able to limit this vaccine hesitancy, because most of our communities are places where we are endemic with conditions. And so if we hesitate this vaccine, then that could be probably detrimental to us. And therefore, we need to be involved in vaccine development and communicating to our people our findings to ensure that they know that what we cook or what we were involved in cooking, they can really rely on that. And again, to inform policymakers to be very transparent with the data they have, and and that's very crucial, because if people can see data and it's very transparent with the processes, then there could be trust rather than the mistrust with the hesitancy. Again, in terms of vaccine development, an area maybe I want to mention will be delivery. I think we can see that delivery has evolved from walking, using taxis, Uber, and all that. But with vaccine development, our delivery has stuck. When you talk of vaccine, all you think about is syringe and needles, and that could probably have affected um, uh, people willingness to take vaccines. And therefore, it's important that we need to start thinking, brainstorming on ways we could help improve vaccine delivery as a Black and ethnic minority. Nanotechnology could be a way to look at where we can have patches. We could start exploring the mucosa delivery mechanism, with the exception of polio, which we could do oral delivery. I'm not sure there is any vaccine with regards to that. So, having worked on meningitis and COVID, it was interesting to also see a dynamic in Ghana, as the last speaker mentioned, that uh, sometimes certain condition comes and we pay less attention to things that are endemic. It was beautiful. We survived COVID. It was nice. In Africa, it was important. The vaccine against meningitis was developed, but within the same period where the global pandemic was occurring, there was also a meningitis outbreak in Upper West region of Ghana, which I wrote on, and there was later a publication on the same issue. So it does show that we need to look at infection holistically, and in the proposal I make, that in future we could have multiple epidemics, because this could be a case. If the meningitis situation had escalated, we would have been managing two epidemics or pandemics at the same time, and therefore we need to start thinking of how we would be able to push forward with vaccine development and to be able to curb such epidemics or pandemics uh, within ourselves. Um, I did also work on other microbes, but again, we do know in most of our developing countries, uh, we are not only faced by bacterial or viral infection. History has shown that we have been successful in developing vaccines against bacterial and viruses. We have, however, paid less attention to fungal and parasitic infection, and I think within the Black and ethnic minority community, it will be important for us to start looking at ways to start developing vaccine that would be useful to our communities. And that also involves studying our own people and having modules that mimics our societies and our communities, so that whatever vaccine or whatever food we develop, it will be good for our communities. Because if you can cook food, but if the food is not ideal for your community, then there might be a reaction that is some of the things we do see in terms of side effect or reaction following individuals receiving vaccines. So I would like to thank all my collaborators in Ghana, at the University of Nottingham, and at uh, Greenwich. I'm
Greenwich.
yet to start
I'm going
my
to start
research
my research
at Greenwich,
at Greenwich.
and thank you all
And
for
thank you
listening.
all for listening.
Okay, thank you very much, Dr. Husman. Your section was really mind blowing, and a particular takeaway for me is that vaccine development is like cooking food. Vaccine development, we can use it, that is synonymous to food, uh, um, food, food cooking. So if you cook, somebody must eat. So when you develop vaccine, somebody must take it. It must actually be taken by people. So the taking process and your stories of your motivation towards going into vaccine development for meningitis and all is really, really remarkable. So do we have any question? Do we have any question? Okay. So in the absence of no question, let me just quickly make this particular comment a question, Dr. Usman. Talking about meningitis and your work with COVID. I could see that you're an African scientist and you really, really understand the terrain of how things.
All right. Yeah. Yeah. Whilst we wait for Sean, I think Medina asked, could you comment on recent effort to build vaccine manufacturing facilities in Africa? I think it's a laudable idea. My experience working in Africa is that I hope it's not just lip services, and the continent needs to make a sustainable commitment in ensuring that it becomes a reality. And looking at science as not um, politics, and therefore should consider engaging people who have the right expertise rather than just family and friends. So I think if we look at it as food, and it doesn't matter, you want to get the best person to cook your food. If we look at it that way, then we might be successful. But if we still go with the favoritism, and looking for people to get round specs in round holes just to get remuneration and title, then it will not be a success.
Yeah, thank you very much, Wayne. Thank you very much, Dr. Osman. So I'm sorry for the initial break. Okay, here is my question. Considering vaccine development as food, and you all know before you can cook a food or cook foods, you have to have ingredients. Like you have to have essential ingredient for the food cooking process. So, what do you say to the entire overall process of vaccine development in Africa, considering the raw materials, the raw ingredients for vaccine development? How sustainable is this? How sustainable is this in the context of Africa? Because I think the last publication I read concerning WHO and Gavi trying to develop mRNA vaccine or in Africa was around 2021, and I don't think it's yet even operational till now, or maybe just kickstart operation. So how do we get these ingredients? How do we get these raw materials so that we will not just have a vaccine development hub in Africa while we still have to keep waiting on the materials and the ingredients for the development? So, what is your overall take, or what's the overall take on the concept of vaccine development in the context of Africa?
Yeah.
Thank you.
All right. Yeah. Thank you very much. Yeah. Thank you very much. I think it's a very good question, and that is what I started to work on. I think before we can think of um, cooking, we need to get the raw materials. And to get the raw materials, currently Africa is lagging. If you listen to Daniel's talk, it was more of genomics. Um, so we need to characterize our indigenous. We need to know what is the monocyte transcriptomics of Africans. We need to understand the transcriptomics of the pathogens that affect Africans. We need to know what genes, uh, what's the genome of Africans. So not until we understand these fundamentals, then whatever vaccine development program in Africa is just going to be a facility. The ingredient will be brought from somewhere, and we just have maybe the kitchen to prepare it. So we need to understand the indigenous before we can think of a sustainable vaccine development strategy.
Yeah, thank you very much. Yeah, I feel that is the right word. We have to be indigenous. We have to take our people, the genomics of our people, or as one of the major raw materials in developing um vaccine so that we do not keep moving around the cycle. So thank you very much, Dr. Usman. Thank you for joining him. And we'll move straight next to our speaker. Our next speaker for this section will be Dr. Lamar Thomas, which is probably going to be our last speaker for this section. So Dr. Lamar is an NIH RACDA postdoctoral fellow at the University of California, San Diego, in the Department of Pediatrics, where she studies microbiology and immunology in the context of host pathogen interactions. Earlier this year, Lama obtained a PhD at Brigham University in the upstate New York in the same field. Her current research is geared towards developing a vaccine for strep throat infections in children and vaginal strep in women. So over the years, she has secured many fellowships such as the HENA HF31 and academic awards for her contribution to science. She's also highly published, producing at least 5 manuscripts in just a PhD program. If you're interested in acquiring similar fellowship award, please reach out to her for advice and guidance. It's good to have you on board, Dr. Lama.
Okay, thank you. All right. All right. Thanks to the organizers of Black in Immuno for like extending this invitation. I greatly appreciate it. Now, as Sian just said, I recently graduated from Binghamton University with my PhD, so I'm very a fresh postdoctoral fellow, I would like to say. So what I'm going to share here with you today is just another scope of immunology, more from a developmental or characterization type of approach. And this is pretty much some of the research that I have done throughout my PhD. And most of what I'll show you today was done in cooperation with my mentor then, which is Dr. Laura Cook. And I'm currently under the mentorship of Dr. Victor Nizet at UC San Diego School of Pediatrics. All right. So as Sion said, like most of my research is centered around host pathogen interaction, but with a particular focus on a vaginal microenvironment. So throughout a woman's life, the vaginal microbiota varies, right? In the reproductive stage, one can have a healthy microbiota, which in humans is normally dominated by generally one organism, such as lactobacillus. However, you can have a transition into an infectious state, right? And in this state, the microenvironment is dominated by multiple organisms, such as some STIs, chlamydia, Gardnerella, trichomonas, some of these examples. Now, pathogens use quite different virulence factors to ensure successful colonization or, infection. And some of these virulence factors include features such as the ability for the bacteria to adhere to the host cells, the ability for invasion, either induced endocytosis or by active penetration. And you'll find that oftentimes these bacteria will secrete some type of enzymes or toxins to aid in its invasion or colonization. They also use the cells to acquire nutrients. And for most importantly, especially for this report, is that you will often find bacteria trying to evade the immune system. All right, so these are just some of the basic virulence factors. Now, Streptococcus agalactiae is also an important virulent pathogen. It is often called group B strep or simply GBS, and it is a gram-positive bacteria. It ranges between 0.6 to 1.2 micrometer in diameter, and they're often formed in chains, which goes by the name coccus. But most importantly for my research is the fact that GBS is an opportunistic pathogen in both humans and animals. Now, an example of GBS as an opportunistic pathogen is seen in vaginal colonization. So generally, GBS colonizes the vagina in a commensal manner, right? However, upon pregnancy, with all the changes in hormones and all the chemical, like there's a whole range of things that goes on when someone becomes pregnant. So what we see is a transition of GBS as a commensal organism to a pathogenic entity. And with this pathogenicity, you can have the transfer or transmission of the bacteria from the mother to the child, either through vertical transmission, or you can also have horizontal transmission, which is seen primarily during breastfeeding. Now, once you have the transmission of the bacteria to the child or the neonate can develop a host of neonatal GBS infections, which include but are not limited to meningitis, pneumonia, and it can, meningitis can result in impaired cognitive function. And in some cases, you can also have the death of the newborn, or even a preterm labor, resulting in the death of the newborn as well. Now, generally, women are tested for GBS or screened for GBS during their 36 to 37 week of pregnancy. And once they're detected to be positive for GBS, they are treated with intrapartum antibiotics. And the most common one is penicillin. But with the use of these IAP, intrapartum antibiotics, there are quite a number of pitfalls, some of which are listed here, but are not limited to these. You can have like a decrease in the GBS. There has been a decrease in GBS early onset diseases, and these are symptom presenting within the first 7 days of life. But there has been no decrease in the late onset diseases, such as those that one will get from like breastfeeding their child. And there has been no decrease in, just the prevalence or the load of the bacteria in a maternal woman. No, studies have shown also that there is quite a decrease in neonatal gut microbiota diversity and abundance, especially with bifidobacterium, when parents or even children are treated with these antibiotics. And with this decrease in diversity in the gut microbiota, what we often see is an increase in the more common bacteria such as E. coli, right? And this in itself creates like a whole range of other problems. So what researchers are now geared towards, oh, did I miss something? Okay, so what we should have here is the benefits of using vaccine development. And of course, we're immunologists, so we know how cost effective this would be, the ease of administration, whether it be orally or, by injection. But more importantly is that it will prevent colonization and decrease maternal diseases, and thus would result in, the reduction in transmission of the bacteria. Now, conventionally, researchers use a capsular polysaccharide to create these vaccines. But now more research is being geared towards a protein component or the protein conjugated vaccine, which they're using components such as adhesins or other microbial agents to increase the immunogenicity of these vaccines that they are created. Now, GBS since 2019, I think the World Health Organization has placed this bacteria on the list of high priority to create vaccines because the effect of the bacteria is so devastating, especially in developing countries, particularly Africa, where you have at least a 30% infection rate and transmission rate, which is the highest that we have seen globally. Now, down to the nitty-gritty. So, like, if we were to implement a conjugated protein vaccine for GBS, what qualities would we look for in a good vaccine candidate? Well, first of all, the protein needs to have an high expression in vivo, right? And that is during vaginal colonization. Now, whatever protein that is, it needs to be conserved in all strains of the bacteria, in this case, GBS, right? So it cannot be serotype dependent. And when we say strains, you have, for example, GBS, they're like at least 1,000 different strains, right? And these strains are sometimes genetically different and more so phenotypically different, where they require different growing conditions and stuff. So we do not want that. This protein also needs to be specific to GBS or specific to whatever bacteria it is that you're trying to create that vaccine for. It needs to be immunogenic, and the antibodies that are produced against it need to be protective. That is, you're getting at least some level of T cell stimulation to give us that memory cell secretion and development. Okay, so in assessing, and what I'm explaining here is basically the first steps that any industrial manufacturing company has to take in determining what would make a good vaccine candidate. I know in the grand scheme of things, we are used to seeing the end product or the production line of things. But before we can even get to the production line, we have to identify something about whatever it is that we're trying to make a vaccine for, what we can use as a target, to create that vaccine. And this is what I am explaining here, just to clear that up a little bit. Okay, so like if we were to find a candidate, a vaccine candidate, one of the first things that you have to do is to conduct a differential gene expression. And this is normally done by an RNA-seq analysis. Now, the work that I'm showing you here was conducted by Dr. Laura Cook, and she studied the differential gene expression of GBS when grown in vivo using a mice colonization model versus GBS grown in the lab in vivo, which is under normal culture medium. And what she observed is shown here on this volcano plot. On the x-axis is the log2 fold change, and on the y-axis is a negative log 10 FDR, which is synonymous to a p-value. So anything greater than 0.05 is considered significant. But what I want to draw your attention to is this gene right here, which is called SAC1753. It was found to be the most highly upregulated gene. Well, technically, it was roughly like a 3,000-fold increase when grown in vivo, like in the mice, versus when grown in the lab. And this sparked your interest because why would you have such high upregulation in vivo of this one particular gene compared when grown in the lab? So we decided that we needed to explore what exactly this gene is doing. Now, upon exploring that and getting some answers to some of our questions, we have since renamed that gene SAC1753 to BVAP, which is abbreviated for Group B Streptococcus vaginal adhesion protein. Now, the research that I'll show you from here on out will be in reference to this gene BVAP. Okay, so just a general idea of BFAP, the protein. So BFAP is composed of 307 amino acids. And what is interesting about this protein is that it has these repeats of 53 amino acids. So if you look here in this diagram on the top in blue, we have what we call the signal sequence. And then in black, we have 5 repeated domains of the same sequence. All right. And then what we observed is that with these repeated domains, you have variations depending on the strains. So this variation, you can have as little as 1 repeated domain or as much as 6. Well, that is what we have found thus far. And what is even more interesting about this protein is that we have run it through a number of different prediction softwares, and we are yet to find any homologous or similar protein with families or domains. So this protein pretty much stands on its own with nothing much that we can compare it to. So one of the first things I had to do when working with this protein was to, of course, create a mutant strain. And in creating this mutant strain, we conducted what is called a sedimentation assay, which is pretty much a measurement of how fast the bacteria settles to the bottom, in this case, of a tube. So if we look at it macroscopically, which is on these images, before we have our 4 strains, which is what we use throughout the slides. You have the wild type strain, which is A909, the mutant strain, which have no level of BFAP expression, the overexpression strain, where BFAP is under the regulation of a REC-A promoter, and then a wild type complementary strain. And what we see before is that, the heterogeneity of the culture, equal distribution of the bacteria in the culture media. But after 900 minutes, what we observe is the complete sedimentation of the mutant bacteria. And when we compare the mutant bacteria to the other strains, we see that they're still pretty much in a heterogenic culture. So with this, we determined that it is possible that in removing this protein from the bacteria, that we have created a hyperaggregation phenotype. And interestingly enough, is that this type of phenotype is one of the sought after characteristics of proteins that are used in vaccine development. So of course, this really got our sparks flying. We're like, okay, we're onto something good. Okay, so from here, we wonder why exactly are we getting this hyperaggregation phenotype? Is it related to the cells themselves? Or have we changed the biochemical, arrangement of the bacteria altogether? So to answer this, we first examined the bacterial strains under light microscopy, just using your basic crystal violet, and we stained our cells. And just off the bat, we can observe that the mutant strain was forming longer bacterial chains when we compare that to the wild type. And when we quantify that, we see where, that's where the mutant strain is twice as long as the wild type, and the complement strain was able to restore that phenotype. So here we're seeing where we have a hyperaggregation phenotype that is being formed, and this phenotype may likely be as a result of this increase in chain length. But then, if you read the literature would suggest that this increase in chain length could also be as a result of changes in what's going on the cell surface or the cell shape. So we dig a little further and conducted some scanning electron microscopy with our strains. And what we observed was quite startling, in that when we look at our mutant bacterial strains under the microscope, we see where the cell shape has changed. It has become more rounded rather than ellipsoidal. Like when we look at the mutant, it's very oval, but here we have these cells looking quite pistachio shaped. So this in itself will lend to like a whole range of other questions, like why exactly as the cell shape change as a result of removing this protein. But what this results altogether tells us is just how important that protein is to the functionality as well as the morphology of the bacterium. So to just get a better understanding of the functionality of the protein, we conducted what we call a adhesion assay. And adhesion assay is pretty much what the name suggests. Like we're testing the ability of the bacteria to adhere to certain components. And we know that adhesion is like one of the initial steps of colonization and infection of any bacteria, well, any microorganism, whether it be bacteria or viruses. You have to be able to adhere to whatever tissue it is that you want to colonize. So we tested or bacterial strains or wild type or mutant and or overexpression. So wild type is in blue, mutant is in orange, and or overexpression strain is in red. And we first tested them on some extracellular matrix components. And these components are often believed to be intermediates between the bacteria and the host cell. So they facilitate the adhesion and colonization of that tissue. And we tested quite a number of these components, particularly fibrinogen, collagen 1 and 4, fibronectin, laminin, and plasminogen. And what we observed was the ability for the bacteria to adhere significantly better with collagen 1 as well as with fibronectin. And when we conducted this on our cell line of vaginal epithelial cells, abbreviated VK2, we saw a similar pattern with the adhesive ability of this bacteria to these cells, right? Where on the x-axis we have our strains, wild type, mutant, and the overexpression. And on the x-axis, the percentage adherence, which is relative to the wild type. And what we see is that in the absence, when we have removed that BFAP or protein from the bacteria, it has lost its ability to adhere to the vaginal epithelial cells, very similar to what we observed in the extracellular matrix component. So in observing this phenotype, we wanted to then carry it over into an in vivo colonization model to determine if that phenotype would stay true. So in this in vivo colonization model, we typically inject the mice with beta estradiol, which is like an estrogen. It serves to pretty much just synchronize the estrous cycle of all the mice. So it's somewhat of like a control method. Then on day 0, we would inoculate the mice intravaginally with 10 to the 7 CFU of bacteria. Then on days 2, 3, and 5, we would collect the vaginal lavage, and then we quantify the number of bacteria that we have collected on agar plates. So here we have the results, which is shown on this graph, where on the x-axis, we have the days where the lavage were collected, and on the y-axis, the bacterial load in CFU per mouse. The blue dots represent our wild type, and the orange dots or red dots represent the mutant strain. And what we see here is that when we inoculate the mice, well, mice that were inoculated with the mutant strain, we recovered far less bacterial load than mice inoculated with the wild type, which indicates to us that these bacteria, these mutant bacteria, have now lost their ability to some extent to efficiently colonize the vaginal tract of these mice. So with this, we then went to understand like where exactly is this protein located on the bacteria, because if it is on the surface of the bacteria, then that would lend itself into an even better story for it to be a vaccine candidate, because then it will be very easy to access. So to understand the position or the location of the protein, we conducted what is called Western blots, and we isolated different sections of the bacterial cell: the secreted portion, the cell wall portion, and the cytosolic portion. And we were able to observe the protein being secreted as well as being a part of the cell wall and the cell membrane of the bacteria. So once again, here we're seeing where not only is this protein being involved in these functions like the adherence to the vaginal tract, but it is also on the cell surface, which is pretty much ideal for any vaccine development or vaccine candidacy testing. So just to bring everything to a close, so today I've shown you like just how we have gone about determining proteins that might play an essential role in a bacterial colonization, and how we can determine if these proteins are worthwhile to carry on for vaccine investigation or development. So we have shown that that the protein has high expression in vivo. It is conserved. Well, I've not really shown much, but it is conserved in all strains of GBS. I have done quite a number of like bioinformatics data on that, which was insane. That's a whole nother story. I would love to share maybe another time. And the protein is also specific to GBS, but we have ongoing research that is currently looking at exactly what are well the quantification of the expression doing in vivo, right? I think we've only done in vitro thus far, and more research is being done to examine the immunogenic potential of the protein as well as the antibodies against it to determine if they will form a protective antibodies. Yeah, I guess. So with that, I would like to acknowledge the Binghamton Biofilm Research Center, which is pretty much everyone here. Not so much of a big group, but a lovely group. My mentor, Dr. Laura Cook, and the members of the Cook Lab, and of course my funding agents, NIH, and the graduate school at Binghamton University. So with that, I would take any questions. And thank you so much.
So Lama, it was such a really wonderful and wonderful section, but we're so sorry. At the interest of time, we won't be able to take any questions for your section. So we do implore you that you join the mainstream, and the other candidate questions that are there, just help us attend to it. Because we're moving to another section now, which is
Okay.
the AI and ML. So we're so sorry for that. So thank you so much, all our speakers, for joining, Dr. Lama, Dr. Usman, Dr. Sarah, and Professor Daniel. So it's such a great one having you on board. And also thank you for coming to share your work. Thank you for the attendees for your question. We do hope that you stick around for our next panel section on AI and ML that is on artificial intelligence and machine learning in immunology. Thank you.